Hi, welcome back. In this section of the immune system, let's learn about different types of interactions between antibodies and antigens. As you can see from this slide, there are a lot of different types. So once I've gone through all of them, come on back and look at the pictures and see if you can talk yourself through each process. So what we know so far about antibodies is that antibodies are Y-shaped and one end attaches to the antigen and the other end can attach to the cell. And so we know antibodies attach to the antigen. And once they've done that, then that antigen can't enter another cell. But the question is, what else happens? There's more to it than this because we haven't gotten rid of the antigen yet. And remember, that antigen could be an antigen on the surface of another cell. So that cell is still alive. And so we have to be able to deal with that cell and kill it. So these next two terms are terms that we learn about when we talk about blood typing and blood transfusion reactions. And the first one is used in normal blood typing, and that's called agglutination. When we take the blood and then we take the anti-A, anti-B, anti-G antibodies, and we see if there is a reaction where we actually can see clumped red blood cells. So that is agglutination, where the antibody is binding to the antigen and causing clumping. And the reason why it's causing clumping is because each antibody can bind to more than one antigen, so you get this big clump. Now, the difference between agglutination and the next process is that in agglutination, those clumps are still within the liquid. In precipitation, they fall out of solution and they sit there on the bottom. And so it's just one step further than agglutination. So they're related but distinct from each other. Now, the rest of the antibody effects, we haven't talked about them before. So pay attention to these because they're quite interesting. The next is something called neutralization, which is kind of what it sounds like. It neutralizes the enemy being the antigen. And the way it does it is the antibodies go and they attach, enough antibodies attach to the antigen so as to coat the active region of the antigen. Does it have to coat the entire thing? No, just whatever the antigenic, the active part is. And by coating that area, that inactivates the antigen, thereby neutralizing it. And another side effect of this is this neutralized antigen then has enhanced phagocytosis because it has all these antibodies on it, which is like putting this little neon light saying, hey, Mr. Macrophage, can you see it now? All right. The next type is opsonization, which is very similar to neutralization, but it doesn't neutralize it. What happens is the antibody binds to the antigen and the macrophage binds to the other end of the antibody. So if you notice, the short arm area, the FC area, is bound to the antigen, and the Fab area, or I said those backwards, are bound to the macrophage. And so since the macrophage has bound to the antibody, which is bound to the antigen, then it's like, it's on the plate right in front of the macrophage and it's going dinner, buffet, snack, midnight snack, it doesn't matter. It's going to have much more efficient phagocytosis because it's right there and it can see it. So here's an example showing that we have the antigen, on this case, attached to a bacterium, attached at one end, the Fab end, and then the FC portion of the antibody can attach to various cells. In this case, is showing it a macrophage, a phagocyte. Now, continuing on after those four specific effects, the next effect is called complement fixation, sometimes called complement activation, but complement fixation is the real term for it. But basically what's happening here is it's activating complement through the classic pathway. So what happens is we have the antibody attached to the antigen, and that activates complement. And so we get the whole C1 to C2, the C4, blah, blah, blah. And then we get the C5, 6, 7, 8 membrane attack complex formed. And then the membrane attack complex is formed on the actual pathogen that the antigen was binding to the antibody. So it's right there next to it. And then it does the usual thing that you learned about membrane attack complexes do. It's going to make holes in it, sodium water going to rush it, and it's going to burst and die. All right. So now the sixth and final one is called anti-dependent cellular cytotoxicity. And if I have it on a later slide, it's going to say ADCC, just to let you know, because this is just a lot of words to write. So here's how antibody-dependent cellular cytotoxicity. In other words, the cell is not going to die unless there's an antibody involved. So we have the cell which has the antibody attached to it, and then the other end of the antibody gets attached to the monocyte becoming a macrophage or a natural killer cell. Okay, so the antibody once again is between the antigen and the macrophage or natural killer cell. Only in this case, it's not going to engulf it. What's going to happen is the natural killer cell or the macrophage is going to be releasing chemical signals which diffuse across to where the antigen containing cell is, and that causes apoptosis of that target cell. So yes, individual cells will die, but basically it can kind of cause a little killing zone going on there. So those are the 6 various types of antibody effects I wanted to talk about. So if you have any questions, be sure to ask. Otherwise, I will see you in class for our next class. Thanks for all your work.